It's a pleasure to launch again the NCCR Distinguished Lecture Series. Today it's my pleasure to welcome Professor Sally Price from University of College London. And for those who don't know, she will give two talks, one today, and she's also our invited speaker tomorrow in the context of the fall meeting of the Swiss Chemical Society in the computational chemistry session in the morning. So Sally is a fellow of the Royal Society. She is a world leader in the field of crystal structure prediction of organic molecules. And as you can imagine, she has a lot of collaboration with experimentalists and with industry, pharmaceutical industry in particular. Her group has been developing methods, advanced force field, interatomic potential, and not only methods but also infrastructure for the accurate modeling of organic crystal structure. She has been leading several projects on this topic, and she also participated with her group in the Cambridge Crystallographic Database blind test. And today she will be especially especially insist on some of the most challenging topics, namely crystallization of polymorph and of chiral molecules.
Thank you, and thanks for the invitation to talk. I've had a fascinating morning talking to people from the Marvel project, and I hope to give you some idea of how things are similar, have moved on, and how they differ between different types of materials. It's now over 15 years since a group of us were able to persuade the UK funding body that a computational method of predicting organic crystal structures would be a basic technology of wide application in science. And they funded us to work with a huge range of experimentalists on this problem. And arguably, we're nearly getting to where we thought we'd be after 4 years. But it's thrown up a lot of very interesting things. So the number of collaborators is far greater than I could possibly list. That website has the papers, and any references in blue, take that as an acknowledgement all the people who were involved in that particular project. So, pharmaceuticals, we moved on from fundamental funding, and recently I've actually been working with Eli Lilly, based in Indianapolis, on applying the methods to pharmaceuticals, very small pharmaceuticals, but that's the industrial application. This shows you why it's so important for pharmaceuticals. That's a transdermal patch for dealing with Parkinson's disease, which gives you you've got the shakes. So that patch is the ideal way of transmitting that molecule into the patient. And what you can see there is that the molecule has started crystallizing in a new polymorph that's less soluble than the ones they knew about when they designed this patch. And obviously having the molecule in the crystalline state renders it ineffective. Pharmaceuticals, you have to think about as having a variety of organic functional groups. Some of them are rigid and bulky. They're joined together by flexible bits, so you've got a lot of different possible intermolecular interactions, and the molecule can adopt an awful lot of awkward shapes. So in the project, we've now got in our database with the structures archiving studies of over 200 different molecules, some of which are two components. But I'd say that this is only at the best indicative of trends. We haven't got much in the way of generalizations. It is heavily biased towards small rigid molecules because they were the easiest to compute, and heavily biased to those where it is known that they can crystallize and there is a crystal structure available. Often the database contains the studies of all the computer-generated structures with a couple of different methods. And recently we're starting to revisit cases such as paracetamol, where we predicted a structure that was later found as a new polymorph with more modern methods. We update it with new polymorphs as they are found, sometimes during the study that's being done jointly with the experimentalist. And tomorrow I'll be talking about how some of the polymorphs have been inspired. The experiment to find them has been inspired by CSP. But the point is that crystallization is very molecule dependent. There are a lot of different stories, and I'll just be taking a few to illustrate pharmaceuticals to you, and that the theory is still very much evolving. Crystal structure prediction methods got their name in the 1990s when they wanted a theory for predicting the crystal structure. Those were the days when the crystal structure was the first crystal that the organic chemist could grow that was a suitable quality for the diffraction experiment, so they could prove that they'd made the molecule that they were trying to synthesize. And so if you're looking for the crystal structure, it is reasonable to assume that it will be the most thermodynamically stable possible structure. And that's all the theory that is coded and done by the computer. We approximate our thermodynamic stability by modeling infinite perfect crystals that are absolutely static, i.e. 0 Kelvin, no 0-point motion, i.e. the lattice energy, the energy to split up your crystal into the molecules which will then in their lowest energy conformation. And this is possible by computer to implement this theory if we can search through all possible crystal structures. Now we can typically search through 1 million with only one molecule in the asymmetric unit and the rest of the molecules being generated by symmetry, and still be not satisfied that we've got a complete enough search. And also we then need to find the most stable, and we now know that we are either using electronic structure theory on a large number of the low energy structures by more approximate methods on each crystal structure, or the methods that have come from the rigid molecules will be doing electronic structure calculations on the molecule in every conformation to get an accurate enough model for the intermolecular forces. So what comes out of these? Well, these diagrams you'll see a lot of. Every point represents a crystal structure where we have the file of all the coordinates in that, so you could go on and calculate a lot of other properties of the crystal structure. Each point is a perfectly valid local minimum, and we expect energy is going down, so we expect it to be the most stable crystal structure, which is indicated by the red symbol when it is corresponding to the experimental crystal structure. And they're usually spread out by some measure of density. Here it's packing coefficient, and hard spheres adopt a 74% packing coefficient. What's unusual about these two examples is the size of the energy gap between the observed structure and any others. This is isocaffeine, and the other molecule is strychnine. And these are unusual in the size of the energy gap, conversion factor up there. And we now understand that because to have a large energy gap means that you've got to have a strongly preferred way of packing defining all 3 dimensions of your crystal structure. And this is unusual in molecules which have been chosen for other reasons than their structure. The complication is the issue of polymorphism. Since the 1990s, the question has changed from what structures, crystals or molecules are polymorphic to what aren't polymorphic. Polymorphism is when you've got the different crystal structures containing exactly the same molecule with the same covalent bonds. It's well illustrated by this molecule. All these crystal structures contain this molecule with those covalent bonds only. And here it's nice and easy to see that the colour and the morphology, the shape of the crystals, is very different. The polymorph can affect all the physical properties. For the pharmaceutical industry, they're obviously most interested in the solubility and the dissolution rate of the different polymorphs, but virtually all the factors, such as morphology, can affect your processing, the design of the processes, your quality control, and also you're only licensed to be delivered in one particular patented polymorphic form by the FDA because of this variation of how your pharmaceutical product will act with the polymorph it contains. So obviously the pharmaceutical industry spent a lot of time trying to avoid problems like the ritonogen patch, of trying to do enough experiments to check that they know all possible polymorphs of a structure. But there are cases such as ritonogen and others where it turns up rather late in the process of developing the pharmaceutical into a delivered product. And a recent survey comparing the results from 2 different pharmaceutical industries on the incidence of polymorphism, depending on your definition, the majority, if not the vast majority, of the active pharmaceutical ingredients are polymorphic. And doing the calculations on the small enough crystal structures, the energy differences between polymorphs are usually very small. They get a bit larger when you've got very different conformations in the 2 polymorphs. So if we compare this with the results of some crystal structure prediction survey done there, you can see the red here are the ones where you've got a characterized crystal structure, and yes, you've got the most stable at the bottom. But you've got, in those black spots, an awful lot of other candidates for the crystal structures where they haven't yet got a crystal structure, and some of these more crystal structures are being solved since. So we've got there that we're generating rather a lot of crystal structures, and we're appearing to over-predict polymorphism if you think that all these distinct crystal structures could be seen as polymorphs. How many of them are artifacts of our approximations in the calculations of the thermodynamics? Particularly the neglect of temperature, because many lattice energy minima may not be free energy minima. Remember, for organics, ambient temperature, we're closer to the melting point of most of these systems than we are to the 0K of our static crystal structure. And so you may have whole functional groups moving a lot, or even the whole molecules rotating, averaging over a different set of structures. Then the other alternative, which the industry is interested in, is it a polymorph that has not yet been found because the right experiment to do it has not yet been performed? So thermodynamic stability is very different from kinetic stability. Once you've packed into the crystal structure, and if you can imagine these awkward shapes, molecules, you see that it can be very difficult to transform to another more thermodynamically stable crystal structure once you're packed. It can also be very difficult to nucleate for the first time a very difficult to nucleate crystal structure. So there can be problems that of this apparently stable structure actually being metastable because it has not had the opportunity or experimentally transformed, been seen to transform despite all the experiments they've done, because they haven't nucleated the other form yet. So the main commercial company that does crystal structure prediction calculations for industry, Avant-Garde Materials Simulation, in the recent Faraday discussion on crystal structure prediction, which was just this summer and going to be a very useful discussion on this, presented a paper where they surveyed 41 of their commercial CSP studies, and with an estimate of the errors in their calculation, made this estimate that a large proportion of small molecule drugs that are currently on the market, the experimentally observed most stable polymorph is not the one that they are finding as the most stable structure. So risking the disaster of the ritonogene patch, or there have been other cases, ritonavir was a case where they had to completely reformulate the case when people on the HIV treatments could no longer manufacture the drug that was licensed. So that's one way of looking at these minima. We've also done some surveys with a lot of experimental work and looked at some of the structures that we're generating as the most stable and wondered, is it plausible that they ever could crystallize and get the molecules to rearrange into that form? So you can see that polymorphism really makes a challenge to our theory of what determines how a molecule will crystallize. So there's the academic interest of crystal structure prediction of trying to get a code that actually puts in a real theory of what determines how a molecule crystallize. But then there are practical applications which you're aware of, designing new functional materials. Another is to understand crystal phases at inaccessible conditions. This isn't just to do with the planetary sciences, but for example, in energetic materials, euphemisms for explosives. You can see in understanding what's happening, they want to be able to simulate the explosive material under extremes of pressure and temperature. And the main interest for the pharmaceuticals, but it does hold for any industry that's making molecular materials, is to aid the polymorph screening of finding all the polymorphs, and because you need that to really develop your products because of this link here between the structure, its properties, how you would make it and process it to get it as things, and the performance of your final products. So, our crystal structure prediction methods, we'd now say that we're trying to generate a crystal energy landscape, the set of all the thermodynamically plausible crystal structures for the molecule. But that is going to be limited, we know, from what we put into our calculations. The assumed molecular connectivity and stoichiometry. Protons are unlikely to be able to jump if you've got a different tautomer, or you're converting from a salt to a co-crystal. You've chosen how many molecules you've got in the asymmetric unit cell, the range of your space groups. It may be less obvious to you whether you're limiting your search to the conformational space that can be adopted of the molecule. We're neglecting thermal effects, and actually there are an awful lot of other approximations in what we're doing where we're relying on cancellation of errors in our relative energy calculations. We don't really know at what level of metastability we should cut off from being a plausible polymorph, and we're always limited by our computer and human time available for doing the work. Nonetheless, it is being effective at being used to design new molecular materials. This is the idea that you use crystal structure prediction and calculate the functional property for the most stable and all plausible polymorphs to see whether it's worth the bother of synthesizing the molecules. This started with CSP started being used for energetic materials, looking to make sure the molecule could pack densely enough, but more sophisticated property structure relationships for nonlinear optical materials, molecular electronics have been evolving. A favourite application is one of the highlights of the work with Graham Day, working for a long time with Andy Cooper's group on porous organic molecules. This is great because it got enough trust in the calculations that they were willing to synthesize a new organic molecule on the strength of the predictions. If you know organic chemists, you'll know how much that means in terms of work. The structure was found. It had the lowest density reported so far from molecular crystal. And here you see every structure they've generated is classified by the dimensionality of the pore for containing the gas molecules. And the one they've got here, naturally found, my pointer isn't, T23A, where the star is highly metastable. That's the structure there looking down the pores. It is a metastable polymorph in this crystal structure prediction, but it comes out because, of course, there's solvent there, stable in the pores, stabilizing it as it crystallizes, but you can exchange the gas or the solvent in the pores. And actually, they'd already published this in Nature before they were eventually able to crystallize the most stable polymorph, which was the lowest energy but denser structure. So this area has two main strands of how people are calculating the final lattice energy at the end of the process. So one is to do electronic structure calculations on each crystal. Density functional theory has helped, but you'll understand that for these size systems, for this number of crystals, we're still rather on the low level of Jacob's ladder of the quality of functionals. Standard is PBE, and that therefore needs a dispersion correction added on. But there are many methods being developed for this, but there's no hope of using these methods in molecular dynamics. The method my group uses, though we may in the end do periodic DFT plus D on selected crystal structures just to see how much the choice of method determines the relative energies, is to do the wave function on the isolated molecule and then analyze this charge density to get a representation of the charge density in terms of charge, dipole, quadrupole, etc. on every atom to represent the lone pairs and pi electrons that determine the directionality of hydrogen bonding, pi-pi stacking, etc. And use that in a model atom intermolecular potential that is anisotropic. Once you're doing conformationally flexible molecules, you have to repeat the calculation for every conformation, and you also have the energy penalty, the difference in the conformational energy that the molecule has gone into a higher energy conformation in the crystal structure in order to improve the intermolecular interactions and so give the lower lattice energy. And we use databases technology to cut down the number of repeat calculations. Usually, in addition to the distributed multipoles, we're usually using empirically fitted explicit repulsion dispersion potentials. But this is an area of research of getting into developing non-empirical methods for all the terms in the potential. So that's development in the theory of intermolecular forces. But we'd also need to be able to develop better models for the intramolecular energy conformational flexibility terms. But that approach has a better prospect of being used for molecular dynamics. But you can see we've all got the sort of tie-in between we can use a better quality charge distribution of the individual molecule than we can if we're doing the whole crystal structure, but you're making approximations in separating the charge density into molecular charge density. So, as Clement mentioned, there are blind tests. This is great for getting industry to buy into how good the theoreticians actually are and their claims. And in the sixth blind test, which is the most recent, these are the target molecules. You're just sent the diagram, but they are good crystal structures within certain limitations. And you can interpret this. This is the number of groups that attempted each structure. So the small, nearly rigid molecule, most 21 groups attempted. The next line is the number of successful predictions, and each group could submit 2 lists of 100 crystal structures. and that's the number that contains that was successful within that list. The next line is the highest rank, including the re-ranking. So first means that someone got it as the most stable of all the crystal structures they're generated. You can see that the small rigid molecule did rather better than the 3-component system or the co-crystal with 2 molecules or this very flexible rigid molecule. But the real star of the show was one of the pharmaceutical industries provided 5 crystal structures from their screening. So we had 5 polymorphs there. This is avant-garde simulations results, which are very good, using, and they're the people that developed using periodic DFT. They initially developed their own dispersion correction for this, for doing that. And the other line, I'm quite proud of how my group did. One member of the group had each target, and for the PhD students, that's a chapter in their thesis, doing this blind test. You have to give in your predicted structures before you get the answer, and then there's a meeting, and the paper therefore has a list of all the different approaches that have been done, and we learn a lot from comparing them. One thing we did learn a lot from the system where there were 5 polymorphs, a number of groups developing DFT plus D methods entered by re-ranking sets of structures because they couldn't generate their own. And they then contributed the energies of the polymorphs there. When there's a dotted line, that's that the group did a free energy estimate, usually harmonic phonons, and those were their 2 lists. And that's the relative energy, and you can see from the symbol that the polymorph is sometimes changing which is the most stable. And aren't they rather embarrassingly all over the shop, given you want to know which is the most stable of those crystal structures, assuming that the company had found the most stable. So we've got a real challenge to the theoreticians to be able to predict relative stabilities. Yep. Accurate thermodynamics. So this is a curve for enantiopure separation. Organic molecules often will have a different mirror image molecule, which has different biological effects. If the word thalidomide means anything to you, that was the worst case example of that. And since when the pharmaceutical industry has had to produce their drugs in an enantiopure, just one hand. And so this is the curve for whether, when you've got the 2 mirror image molecules in your solution, whether you crystallize as a mixture of mechanical mixture of crystals can each contain just one hand, or you have the 2 molecules in the same crystal as a function of the energy differences between the 2 crystals. And you can see it's rather a tight curve. If the lattice energy difference is big enough, you know that you can't do it, but it is very tight there. And, you know, think of the sort of approximations you might make. The ratio of the relative solubilities of the 2 crystals depends on the exponential of the energy differences of the solvation, of the free energy of solution of the 2 crystals. But it's the same molecule. Surely the solutions are the same, or at least non-ideal to the same extent. So it's just to do with the free energy difference between subliming the molecules. Surely all the entropy effects are going to be much the same, so it's just the heat of sublimation difference between the 2 molecules, and surely your 0-point energies and your heat capacity differences are going to be the same, so it's just the lattice energy difference. Trying that out against an experimental group in Max Planck, Magdeburg, on 3 molecules, none of those approximations were good over a range of molecules. And the worrying thing is that that curve's much the same for different polymorphs. It just becomes more symmetrical when you don't have some factors of 2 in. And you can understand that from these calculations. For each of the series, the top in the square are the molecular frequencies, and you can see that some of the molecular frequencies are down in the low energy region of less than 150 centimetres to the -1 to a different extent, and then you've got the frequencies at just k equals 0 modes, phonon modes, they've got different crystals. And so, for instance, here in naproxen, which is a drug molecule, in the racemic form, you've got a very low energy sliding of the two layers across each other, giving you a very low energy frequency there. So it's not surprising that you do have differences in 0-point energies and differences in the heat capacities as a function of temperature between the two structures. So we've got a real challenge here of getting to what we want to get at from crystal structure prediction. Even at the 0th order, when we're dealing with lattice energy, there is a challenge of going up in increasing complexity of your system and the types of different inter- and intramolecular interactions you are trying to balance. That diagram is that drug molecule turned on its side. It's the chloride salt, and you've got layers where the layers can be bridged by different alcohols with the OH bonding to the chloride salt, and you have a nice van der Waals layer of the hydrocarbon chains. That's a dialcohol there. We really should be, for testing our theory, be looking at thermodynamics accuracy. Okay, calculating with pressure is easy. Calculating the effect of temperature isn't easy, but we also know that the different polymorph relative stability can depend on the crystal size, surface versus bulk energy, and the solvents, including the relative humidity of water around. So actually testing the basic theory of thermodynamics is no mean thing to do. And then what do they really want out of a genuine crystal structure prediction program? They don't want what we've got here of over-predicting polymorphs. They want you to just predict the structures that can be found, which implies enough of a knowledge of their properties for a recipe for how you actually find them. So, we've got a long way to go. Okay, now just a few quick examples to give you a sort of flavour of some of the issues in organic polymorphism pharmaceuticals. So here's one drug, tazopholone, which is chiral. You can see the hydrogen there. When we went into it, Eli Lilly had done some screening over a decade ago and knew 2 polymorphs for the racemic form, which had the same hydrogen bonded layer but stacked in different ways. But they'd also done the structure that the enantiomer pure crystal had 2 different conformations in its unit cell. But if you seeded a racemic melt with the enantiomer pure crystal, you got the very worrying solid solution with the same structure. So actually you were absorbing the molecule of the other hand, which is not what you want to do for chiral resolution. So could we suggest that? They were going to do some experiments. First thing they did, pull out of their sample a new polymorph, which was related to the other 2 they knew about, same layer, but different symmetry relationships for the stacking of the layers. There. Try to get our energetics to test our energies. Oops, even by doing the measurements on single crystals, they were getting variations in melting points and heat of sublimation, which eventually, by looking at the raw diffraction data, it's streaky. There's disorder in it. Okay, disorder. So how did we do in CSP? So generated all these crystal structures, which Louise lovingly classified by type of conformation in colour and type of hydrogen bonding motif. And of those that were generated, only a few need to be worried about. One of them, the green squares, are that hydrogen bonded layer in the observed racemic forms, and yippee, the most stable C prime equal 1 structure has been found. The other two are low in energy, but because they're Z prime equal 2, two different molecules, you wouldn't expect to find them. But we've got a few more, one's hiding there, green squares there. So by the fact that we have generated different stackings of this layer, we are warning that different stackings have been generated and they're so close in energy, we are warning that this order is quite likely, that you may be, as you crystallize, getting errors in stacking the crystal structure. Enantiomer pure, Z prime equal 1, just one molecule, you can't get anything, you can get very few low energy crystal structures, and none of them as low as the one they observed, where you've got two different conformations. And when you take the energy diagram over, there is a racemic structure with the same cell, etc., and very similar packing as a racemate there. What's happening for this molecule is that the different conformation can play the same role in the crystal structure as the opposite hand. So that's why you can form the solid solution between the two hands. Okay, this one was done with GSK, but I just wanted to include it to illustrate you've got a lot of diversity in the conformations. Yep, for this fairly simple molecule, you've got 3 different possible hydrogen bonding positions, and you've got a lot of very flexible rings, and you can see for this one case where there's sort of three different areas for that area, you can imagine once you've packed the crystal, you're going to have problems rearranging it. So essentially we did 9 searches varying the other torsion angles there. And we were delighted that the one crystal structure that they knew of came out as the lowest. But we found another structure which is hydrogen bonded, so the intramolecular energy penalty is high, but the hydrogen bonds mean that it's got virtually the same lattice energy. You carry on, the other hydrogen bonding conformation could also be found. Yep, we're generating quite a few different structures that are close in energy and have such different conformations that once you'd found them, it's plausible that it'd be very hard for them to transform back. So they should be long-lived polymorphs if you found them. So they did go away and do a lot more screening work, and they were able to find some metastable solvates, which did have the differences in the hydrogen bonding, but the rest of the conformation was in the same as in the neat polymorph. So in this case, it looked as if the kinetic favorability that the most stable structure crystallized so easily was what was preventing finding any other polymorphs. Hopefully none of you on olanzapine as a drug, which was a big one, Lilly's blockbuster that came off patents just as we did this work with them. But this, again, you've got 2 very different types of conformation, but in all the known forms, it's the same conformation packed as a dimer, which has the hydrogen bonding group sticking out. They didn't know, despite Lilly having this under patent, doing an awful lot of work on the 2 forms, they'd been unable to crystallize forms 2 and 3 separately. They're always found as a mixture. But in our project, and also simultaneously in India, Nangia, were able, out of the mixture of crystallites, to pull out a crystal that was big enough for current methods of solution from single crystals, giving the form 2 structure. So the first question from the CSP was, could we find form 3? Here you can see we've generated an awful lot of crystal structures, and only the red diamonds contain the dimer, But we've got the most stable form, form 2, but there are an awful lot of other rivals structures out there. The big thing is that olanzapine cannot pack densely with itself. Yep, the packing coefficient, maximum 70%. So to try and find farm 3, you look at the powder patterns from all the predicted structures and could find one which was only a kilojoule per mole above form 2 that looked very plausible. It's got, you can see this overlay, essentially the same layer structure, but the layers are stacked in a different way, but it's only how the little hydrocarbon stuff is, weak van der Waals forces, how the hydrocarbons are dealing with. And the two being related by alternate stacking accounts for the concomitant crystallization, maybe cross-nucleation, or is it stacking disorder, but just the complexity of the problems they're having with it. And so, although it's not an absolute kosher model, it looks like a very good explanation for this. And, olanzapine in the survey, they found 60 forms. A lot of them are solvates based on a layer structure which doesn't pack well with itself, and you get different solvents packing in there, raising questions about how olanzapine nucleates and grows. So more recently, we've been looking at the nucleation by AFM, and there's some rather surprising results that if you put olanzapine in water, it will hydrate and form the hydrates. But you could see in the AFM that you're getting hillocks growing at the ledges in the olanzapine surface. If you were stirred conditions, then the droplets detach and form the kinetic metastable dihydrate polymorph. If it's left nice and quietly under the AFM, you can see it crystallize into oriented crystals of the more stable dihydrate polymorph. So we had to look at that, and this has some information about modelling. Yep. First of all, why are we seeing these dense hillocks by the ledges? Well, we tried building a model for the ledges and just letting the olanzapine dimer dock onto it. Obviously, the lowest energy site is carrying on the crystal structure in the ledge site. But there are an awful lot of other structures where the dimer will stick in a low energy minimum that are lower in energy than it going down onto the surface there. So it's not surprising that in the dissolution of the olanzapine and the nucleation of the dihydrate, you're getting these blobs that it's very difficult to orient the molecule in the process of nucleation there. And doing some calculations where you've got perfect surface and then using a nanocrystal of the two different dihydrates above the lattice, we could show that the dihydrate D, as a function of the angle of the nanocrystal, had some preferred orientations which did match with the observed orientation of the dihydrate crystal structure on the olanzapine surface. And there's a better match going down between the surfaces than there is with the kinetic product, where the ones that were seen on the surface probably nucleated elsewhere and had just fallen down in any old position. So, where are we now? All our so-called crystal structure prediction methods are doing is telling us what types of packings are thermodynamically competitive. Occasionally, clear prediction of the crystal structure. Take an isomer, same method, you've got caffeine, and that bundle of crystal structures are just a few of the components of the disordered static structure. So caffeine, the low temperature form, is statically disordered. The high temperature form is nice and symmetrical because the molecules are whizzing around in the plane, and those are just some of the structures that contribute to the disordered structures. It helps you interpret the experiment. The usual position is that you're predicting polymorphs with a challenge to either find them experimentally or eliminate them as being of no concern or interest. Of course, this is an area which is of interest to industry, and we're trying to sell them. So that was a review that we did of all the publications joint with an industrial author where they've done industrial screening on the molecules of a CSP. And yep, it can give you a lot of help with characterizing your new forms and may confirm you've got the most stable structure. But when there are other ones, the question is, can we suggest a non-routine experiment to find the structures that haven't yet been found? Or are these structures artifacts of our static calculations or our just approach? So I hope I've convinced you that if we really want an organic crystal structure prediction that lives up to the name that we've got to do a lot more to understand the kinetics of crystallization, of how we get to the structures that are observed. So it's a field which will go for many more decades. A huge number of people have been involved in this. Some current people with my group and my colleagues at UCL, a lot of work has been done with the Strathclyde group, which have a major centre on organic crystallization, and the group at Eli Lilly. Huge amount is the people who develop the computer algorithms. I've been involved with developing DMA Cris all the time. I've been at UCL and people at Imperial doing the algorithms there. And it's my PhD supervisor, the distributed multipoles, that have been the base of it. CDCC for organising blind tests, and this is a field which is a joint endeavour because you do from the blind tests learn about the different methods, and we've also had considerable funding. And thanks for your attention.
Excellent. It's a great journey in the world of crystal structure. Questions? I start with the first one. So at the end of your talk, you come up with a question, right, in a sense. Are our static computation sufficient, or are the experiments maybe, the experimentalists need to go further and try to find other known state-of-the-art way to crystallize?
So I think this is what I'll be talking about next. It's up to us to find non-standard
way to
crystallizing them. So in industry, there have been developments of different robotic forms of trying to survey thousands of crystallization conditions. Yep, it's easy enough to vary your solvents depending on the range of stability, but cooling rates, rate of agitation affect things. And the big problem is that surfaces can have a major influence on what's formed. Impurities can have a major influence on what's formed.
That's from the experimental point of view, but now from the theoretical point of view, how much do you trust the methods that are viable now? And if you had to improve one aspect, will it be more the accuracy of the lattice energy, or the way you really explore the free energy landscape of the individual molecule, or the crystal energy landscape, which...
So we would now... I've mainly been working jointly with the experimentalists, and it is tended to be a feedback that you use the new polymorphs as they're discovered to benchmark your methods as you're going. But that's why we would often take a set of the rival crystal structures and then do various density functional theory calculations to see how sensitive our relative lattice energies are. And the example I gave there from the blind test is not atypical. You know, the DFT plus D calculations, even with the same functional, can vary a lot on the dispersion correction. If the crystal structures have a variation in density, you'll be very sensitive to that. So you have to do a sort of analysis of how much you believe even the relative lattice energies. And on top of that, we do quite a lot of looking at how classifying the diversity of the structures with some feeling of, well, if they're similar and have motifs in the case, is that likely to be a disorder, or that you'd never get them there because they would have all snuggled down to the most stable of a family? And this is where different methods of analysing the different structures becomes important.
Post-processing.
Yeah, it's the post-processing and knowing what you're looking for of differences, and that's where we're still very much in a learning cycle of that. And in some senses, it is the problem that a lot of things don't crystallise well. That being able to grow the single crystals is actually a sign they are fairly readily crystallised. So often you can only get powder samples, which is where the solid state NMR crystallography and solving powder X-ray diffraction, people putting different methods together. But industry will be able to usually puts an awful lot more resources into knowing it, up to the point where that molecule is still viable as a potential pharmaceutical. And of course, they don't let us onto anything that strong, so the collaborations tend to be on the molecules where the industrial scientist is interested in being able to carry on as a collaboration.
I was intrigued by the results on the molecular crystals. And there you showed a graph where you had energy versus, I think, density that nicely suggested some kind of funnel, as the molecule would drop in there. But why did you plot density on the horizontal axis? Had that, I sort of was wondering, because you can plot it against anything else, and it may not show such a nice funnel. So is there some physical meaning there?
Roughly, I'd say that the tends to get that funnel is just because of the importance of the dispersion interaction. Yeah, as you get denser, you get more dispersion, and so that tends to stabilise things. You do get cases where, of course, the different directionality of the hydrogen bonding compromises with density there, but it has been a traditional thing to do it with, but increasingly we are now, you take the structures and use other dimensions, colour coding, for the property, which can be a structural property or could be the physical property that you're after. But you know, the use of density or packing coefficient, which packing coefficient is less dependent on your actual atoms involved, so it's more comparable, is usually partly by tradition, but if you're not packing densely, that is often a sign that you're likely to have solvate formation. And I think that is one of the things that distinguishes a lot of pharmaceuticals that have ugly shapes, that there's no way they can pack densely, from the simpler molecules that are a relatively simple shape, where there are a variety of ways that are equal in density.
Yeah.
Do you also do simulations of nucleation or?
So I'm interested in the simulations of nucleation, and nucleation is a big topic in this field, both experimentally and computationally. And that's one of the implications of that olanzapine work, is how the surface is influencing which structure is nucleating and growing. So part of the problem is that you very rarely get nucleation as you'd set it up in a computer. Yep. In practice, you usually have, almost always will have surfaces and other molecules present, which are not in the classical MD simulation of just solvent molecules or just the molecules if you're doing nucleation from the melt. And this is where the increasing observation of these two-step nucleations, where things tend to densify first before rearranging a nucleation. That's mainly an experimental observation, but some people are seeing it in MD. That is rather different from a simulation idea based on classical nucleation theory. But there are a lot more problems of MD, or a lot more advantages of MD, and that is actually representing the fact that the molecules are moving. And the motions of the molecules are vital. It isn't showing nucleation, but you've got, and there you're right, you've got that difference between at what stage is the crystal structure set in from there. Yep, that is a big problem of experimentally studying at what point is the nucleation happening. So the classic NMR experiment, that's an acronym. It's an MMR method that can switch between the liquid state and the solid state NMR experiment, which will only detect the solid particles. That has been developed as a means of studying it by Kenneth Harris's group, but even there you're quite large samples by the time you're actually detected by the NMR.
Just wanted to know if there is some resonance in sugar, for instance.
Pardon?
Sugar, you have some experience on sugar?
Sugars are horrible. So many OH groups around, but some of them do crystallize well, and others don't. And yes, sorbitol versus mannitol and the behaviour of different crystallizations. Sugars are often used in a lot of pharmaceuticals as what they call excipients. The pill you take is often packed up with a lot of other things in there. And people are more and more studying the polymorphisms of the sugars, but that can be quite difficult, mainly because of all the flexibility in there.
Have you ever thought about problems where you need to change the shape of the crystals?
You mean the morphology?
Well, the shape. For example, if you want to make membranes with porous media, you would like to have flakes with the pores perpendicular to the plate. But the natural crystals are very different. So you would like to develop methods to steer the growth of the crystals.
Yes. Okay, so there you've got the examples of the different morphologies as a function of polymorph. And yes, there is quite a lot of work being done on trying to develop methods of predicting morphologies. We do, in our database, occasionally do a very simple method that is solvent independent, based on the attachment energy methodology, mainly to look at whether you are expecting an extreme plate or needle, and look at relative growth rates. And one of the advances that is happening is that more and more crystal structures are based on needles or plates. The techniques for solving the structures is improving in those fields, some of which are helped by CSP methods for solving those structures. So at the moment, there is, because of the importance of morphology in processing and other aspects, there is quite a major area of interest of predicting morphologies. And Mike Doherty in University of California, Santa Barbara, has a methodology which is somewhere between a very simple one, but does actually include some effective solvent and isn't quite as extreme as actually trying to do it each face by MD simulation. But the aspect of how does the crystal grow, how do you desolvate and get it in position, it's a challenge. And then you've got the aspect of having your impurities either enhancing or suppressing the growth of a face, and even sometimes stopping the appearance of a face, which is one of the reasons why often the first crystals, the first polymorph that comes out of an impure sample may often be metastable, and it's the impurities that have been influencing how it crystallizes. Because as you can imagine with this in synthesis, you've usually got an awful lot of impurities which are structurally similar chemicals with structurally related that can nicely dock into one face. So it's a whole big new field there.
Yeah, Linda, go ahead and then Linda.
Can I ask which global searching algorithm do you use to find the possible polymorphs?
So Crystal Predictor, which is the one that Imperial are developing that we're using, is based on Sobel sequences. But there are a variety of other methods that are available, some based on simulated annealing, some on genetic algorithms. That's one of the fields where, you know, this is a rich field for all sorts of aspects of computational chemistry, in that in the blind test you'll see that there are quite a lot of different search methods being implemented and tried. And some of the aspect of the success of these does depend on what sort of molecule you're dealing with. If you just want to deal with simple rigid molecules, it's a lot easier than if you're trying to do the pharmaceuticals, where you have to have your conformational as a search variable, because changes in conformation really do change the packing.
So I think it's just going back to some of the first comments. So when we use CSP to connect into structure determination, we're concerned with taking the smallest number of plausible structures. So I noticed on your graph, in several of these graphs, you have plausible structures as a line which is a fixed lattice energy. Graham usually tells us it's 10 kilojoules per mole from the lowest energy that he's predicted, so then it will be a moving target on your scale. And then if I listen to Clemence and Bernd, I'm suddenly thinking, well, why is it a straight line at all on these graphs? Why isn't it a curve, or favouring certain densities or certain other physical? So how do we decide what are the plausible structures that I should consider if I'm trying to determine a structure experimentally?
Very nasty question, because this is where the more families of polymorphs you have, the more experience we get of where the number will cut off. And yet, yes, as you know, they get, in some senses, you feel that as they get more metastable, the driving force to go to the more thermodynamically stable form is increasing exponentially, so they should be less likely. But of course, that's not what determines whether they're long-lived. It's the barrier that they've got to get over. And one of the worrying things is that sort of 10 kilojoule per mole as a sort of maximum rather nicely fits with that graph. But that is would be roughly what you'd get if you're just talking about solution-grown polymorphs. Yep. The distinction between whether or not you could get them from the solubility difference gives you, from that argument, about that amount of leeway, whereas increasingly, both in the pharmaceutical industry, where they desolvate solvates to find new polymorphs, or in this nucleation, two-step nucleation process, where you've got a real dense cluster, but still liquid-like, of your solute with some solvent molecules. You've got a lot of structure finding before you're desolvating somewhere in that process. Those could have potentially larger energy differences. Yep. Certainly, there's a suspicion that if you actually sort of made your polymorph in situ by photochemistry, for example, you might have very large energy differences. So learning what that is going to be a function of the differences in the structures and the differences in the methods that can be applied, which is a horrible conclusion to be coming to.
Well, I'm going to close the question session because we have an aperitif, and if you want to ask more questions, Sally, you're welcome to do so downstairs. So thanks Sally one more time.